Okay. Yeah, so good morning again, everyone. I saw that we have some new students today, so I just wanted to introduce myself again. My name is Debbie Frederick. I'm currently a senior medical lab assistant from St. Lucia. I recently finished my degree in biochemistry and biology this year at the University of the West Indies in St. Augustine. I did 2 years of facilitating STEM-related workshops. Um, like I was speaking to the students yesterday, my passion lies in genetics and really understanding the link between like our genetic makeup and diseases. Um, One of my goals is to launch a biotech company in the Caribbean and really to pave the way for the next generation to get into the field of biology, biochemistry, and biotechnology. So when I saw this opportunity to facilitate a program like this, I quickly jumped on board. Um, but yeah, today we'll be going into immunology, and we'll be speaking about the different components of immunology. This I did an entire semester on immunology, so fitting all the concepts into one session was pretty difficult, but we'll go for it. And I tried to draw out some of the pieces I found to be the most important, well, and fascinating. So let's begin. So what is the immune system? The immune system is a network of cells, tissues, and organs that work together to defend the body against harmful substances. And these would include pathogens, toxins, tumor cells. And for the students that were there yesterday, when we went into medical microbiology, we would have spoken about the viruses, bacteria, and fungi. So these are some pathogens that you would encounter. The immune system has various components. It's very complex. So it includes cells like white blood cells or leukocytes, and these would include neutrophils, natural killer cells, macrophages, eosinophils, just to name a few. There's also lymphocytes. So these will be your B cells and your T cells. And different systems like your lymphatic system that includes your lymph nodes, your lymph and the lymph vessels. Um, and different organs like your thymus, your bone marrow, and your spleen. Your thymus is where your T cells would mature. We won't go into the whole maturity and activation. We won't go in depth, but just for your knowledge, that is where your white blood cells would, your T cells, sorry, would mature. And in the bone marrow is where your B cells mature. And that is also the site where white blood cells are produced. So this is just a summary of all the different cells involved in your immune system. Usually starts with a stem cell that could then go into a lymphoid or myeloid pathway, and depending on which one you get, like various cells. So you go in, if you go for the lymphoid, you get the B cells, and then your B cells could differentiate into plasma cells when they're activated or memory cells. Plus, the role of plasma cells is solely to just produce antibodies. So they would just massively produce antibodies when your body's under attack, and your memory cells are there for secondary response. We'll go into more detail later down in the presentation, but I just wanted to show you just the number of cells involved. And these two set of cells, your T cells and your B cells, they form part of your adaptive immune system. And again, I'll touch on like what exactly that is, but just know that it's like more of a specific response compared to your innate immune system. So then there's the T cells. Your T cells there also has memory T cells. I know for a while in science, we were saying that T cells don't produce memory cells, but recently we've started accepting the fact that there are memory T cells, not only B cells are memory cells. Then there's the cytotoxic T cells, and there's also the T helper cells in your, now something that's really interesting is part of your, like from the lymphoid line is your natural killer cells, but they're not part of the adaptive immunity. They're mainly part of the innate immunity. So that if you ever do immunology, if you ever do an immunology course at the university, that's one of the multiple questions I always see that catches students. But yeah, it's part of your innate immune system, the natural killer cells. I think those cells have the best names in our bodies, just because that's literally what they do. They kill and they call NK cells. But then there's a mast cell, neutrophils, eosinophils, basophils. All these come from the myeloid um, side of from the stem cell. And there's also the monocytes, who could then differentiate into macrophages and dendritic cells. So this is just a summary of the different cells involved in the immune system. So type of immune response, we'll start with the innate. The innate immune response is the first line of defense that provides a rapid, non-specific defense against pathogens. It is present from birth and does not require previous exposure to the pathogen. The key components of the innate immune response include chemical and physical barriers, phagocytes, natural killer cells, for example, like I just said, inflammation and fever. So yeah, the main part about this is non-specific defense. So can anyone name, just give me some examples of a physical or chemical barrier that you know of in the body? Let's see what anyone can think of. Yeah, so physical barrier would be the skin, yes, correct. And any chemical one? Think of acid. Where in your body has acid? Yeah, the stomach acid, yeah, correct. So that's correct. So this is just a summary slide showing you the different parts of the body and some non-specific barriers. So there's lysozymes in your eyes, the removal of particles by the cilia in the nasopharynx, the skin, like Giovanni said, is a physical barrier. Unless you cut your skin, it's very difficult for pathogens to enter just because of the layers it has. With the rapid pH change, that's in the small intestine, because if you think the stomach is very acidic, so it's like pH of 2, very acidic. But when you go into the intestine, the pH increases. So just a change in pH could destroy some pathogens. There's also the mucus lining the trachea, flushing of the urinary tract, and just the normal flora in the large intestine, because the large intestine does have some bacteria. And this is just a table showing you again, just showing you more elements of the innate immune system. It's not all about cells, like I said. So there are atomic barriers like the skin or the mucous membranes. There's also physiologic barriers like temperature. In the session yesterday, I was speaking to the students about the cordyceps fungi that was shown in the show The Last of Us. I'm not sure if any one of you have watched the show or played the game. But essentially, it's about the fact that cordyceps, which are like zombie fungi in nature, what they do is they affect some insects, and they cause them to, like, they take over their brain to have them go to high points in the rainforest where they clinch onto like branches and they die from there. And the fungi is able to grow and disperse its spores from that aerial view. So in The Last of Us, the theory is that these cordyceps, because of climate change, they could now affect um, human beings, and it's about a zombie apocalypse happening where, because of the fungi, we become zombies and are controlled. Um, but for that to happen, it's very difficult because of the temperature of our body. Like, it's so hot inside of us that it's difficult for a lot of pathogens to survive. So just the temperature is one thing. That is why when you have Like when you're infected, one of the symptoms that you get is a fever. That's when our immune system releases some pyrogens that goes to your brain and causes your brain to increase the temperature. And the main reason for increasing the temperature is because it makes it difficult for some pathogens to survive and difficult for them to replicate. It also gives energy to our immune system to fight off the infection. So that's why when we have a fever and we go and we buy medication that like brings down our temperature, it's like we're basically going against our immune system because we should that it's a natural process to increase the temperature to make it difficult for those pathogens to survive. But because like the discomfort of being hot, you tend to go in and get some drugs that bring it down, but that's not always the case. But increasing the temperature can also affect the functioning of the system. Yeah, it does. But the thing is, your enzymes won't break down right away though. Your enzymes won't break. Yes, your body, it's a natural response. Like if you have a fever for a long time, like a prolonged period for weeks, then it will become a problem. But you tend to get a fever like right after an infection. And so maybe and it lasts for about 1 or 2 days. It doesn't last longer than that. If it lasts long, then that is when it becomes a problem. But if you have a fever for 1 or 2 days, it wouldn't be that threatening to your body. Because just so you know, our body has something called heat shock proteins. And what these do is they help proteins to fold. Because if you're I'm going to the biochemistry side of it, but at increased temperature, proteins tend to unfold. That is because a lot of the weak bonds that holds the amino acids together, for example, hydrogen bonds, tend to break, and the protein will unfold. And when a protein unfolds, then it can't really function properly. But we have some enzymes called heat shock proteins, and they're synthesized at high temperatures. And what they do is they assist with folding your proteins back. So we have mechanisms that could counter high temperature. It's just that when it's prolonged, it would become a problem. But good point, Giovanni. Yeah, all that as well. But low pH, obviously, you know how acidic could destroy microorganisms. There's chemical mediators. One I will talk about is the complement system. I don't want to go into detail because it's really complex, but I just want to show you just how complicated our innate immunity is. So it's not only about like barriers. We actually have different proteins in our bodies that could actually um, really do some cool things, like it literally puts a hole in microorganisms. The next one is interferons that helps against viruses. And these are some chemical mediators. The next one I want to touch on is the phagocytes. So that would include things like your macrophages, your neutrophils. And what these do is they would engulf the microorganisms, break them down. And in the case that they're antigen presenting cells or APCs, they would like where they would put the antigen onto their surface to encourage our immune system to recognize them and then further degrade them. Inflammatory barriers. Yeah, so another key thing with an infection is there is a lot of inflammation. Inflammation really allows the body to recruit more immune cells and bring them to the infected area. So yeah, our body is very interesting when it comes to that. So this is a video I have called, it's from Crash Course on YouTube. If you've never come across it, I advise that you know, follow them or watch some of their videos. This video is 9 minutes, so but it's very interesting, just introducing the innate part of the immune system. So it will just reiterate what I've been speaking about. So just have a listen, guys, and then we'll continue after that.
You may not know it, But your body is engaged in a never-ending battle. You are literally covered in staph and strep and E. coli and all sorts of dubious characters that are intent on using you and your body's many resources to feed themselves, find shelter, and reproduce as much as they want. And hey, we all gotta make a living. But it is not your job to give these guys a free lunch. So your body has developed a three-part policy toward these shady customers, and its enforcement is handled by your immune system. The immune system is different from all the other systems we've talked about this year in that it's not a specific tissue organ system kind of system. Instead, it involves a whole bunch of different tissue groups, organ systems, and specialized but widely distributed defense cells. Together, this league of extraordinary substances joins forces to perform all of the defense functions your body depends on to keep you alive in an incredibly germy world. And the first line of defense in this never-ending battle, that's your innate or non-specific defense system. Like your average frontline soldier, it's prepared to immediately engage with anyone suspicious, and it mostly includes stuff we were born with, like the external barricades of your skin and mucous membranes, and internal defenses like phagocytes, antimicrobial proteins, and other attack cells. But some enemies must be fought with special forces, and here your body can deploy your adaptive or specific defense system, which is more like your SEAL Team 6. It takes more time to call in, but it's specially designed to go after specific targets, and it keeps files on those bad guys so it knows how to handle them next time around. But today we're going to focus on your innate system and look at how it uses an arsenal of physical and chemical barriers, killer cells, and even fever to keep you healthy, proving that sometimes the symptoms we associate with illness are actually the signs that we're healing. Just because something is simple doesn't mean that it can't be elegant. I mean, your body is capable of some incredibly sophisticated things, including defending itself from infection. But occasionally, there's something to be said for brute force, and a lot of your innate immune system's functions aren't exactly subtle. For example, your body's very first line of defense is a simple physical barrier, and it works. Like a wall around a fortress, your skin does a fantastic job of keeping out all manner of malevolent microorganisms. As long as that tough, keratinized epithelial membrane doesn't get torn open or busted up too much, you could probably make snowballs out of raw sewage and still be all right. Although, no. Your many mucous membranes also provide a handy physical barrier. You'll remember that they align any cavity that opens up into the germy outside world, including the respiratory, digestive, urinary, and reproductive tracts. Not only do your skin and mucosa supply simple physical protection, they also pack some serious chemical weaponry. Eat some questionable leftovers for lunch? Don't worry, your stomach is literally filled with acid, so you probably are covered. Walk face first into your coworker's nasty sneeze cloud? No worries, your nasal passages can whip up a tissue box worth of sticky mucus to help trap viruses before they enter your lungs. You've also got bacteria-fighting enzymes in your saliva and lacrimal eye fluid, and peptides called defensins in your skin and membranes that help keep bacteria and fungi from setting up shop around inflamed or scraped skin, which no matter how careful you are, you're gonna get one way or another. Maybe you shave with a dull blade, or you just brush your teeth too hard, and don't get me started about the dangers of bagel cutting. So when you've breached that first simple line of defense, it's time to call on your second line of internal innate defenses. This is where your body starts pulling strategic maneuvers like firing up a fever, releasing chemical signals, causing inflammation, or other defensive tactics that help identify and attack infectious invaders. Some of the first defensive cells on the scene are your phagocytes. Their name literally means to eat, and like Pac-Man, they indiscriminately chase down intruders and gobble them up. And they come in a few different varieties. First, you got neutrophils, which are the most abundant type of your white blood cells. They kind of self-destruct after devouring a pathogen, and in fact, you've probably seen piles of their little dead bodies, because that's what pus is made of. But the bigger, tougher phagocytes are the macrophages. They're derived from monocyte white blood cells that have moved out of the bloodstream to occupy tissues. And some are free types that patrol tissues looking for creepers, while others are fixed, attached to fibers in specific organs, devouring anything suspicious that passes by. So when a macrophage, in say the finger I just cut slicing a bagel, sees a new bacterium coming along, it snares it using cytoplasmic extensions, reels it in, completely engulfs it, and essentially digests it and spits the rest out. And unlike neutrophils, it can do this over and over again, like a boss. So not all your defense cells are phagocytic. You've also got cells with what is by far the awesomest name of any cell in the body, the natural killer cells. You can call them NK cells if you want to, but why would you do that? Anyway, these tiny assassins patrol your blood and lymph looking for abnormal cells and are unique in that they can kill your own cells if they are infected with viruses or have become cancerous. How can they tell? A normal, healthy human cell contains a special protein on its surface called MHC1, or major histocompatibility complex, complex, but if it's infected, it stops making that protein. And if an NK cell detects a defective cell, it doesn't swallow it whole like a macrophage. It pokes it with an enzyme that triggers apoptosis, or programmed cell death. Pretty awesome. So those are some ways your innate immune cells handle their enemies, but how do they know where to look in the first place? So let's talk strategery. So say you're in a banana factory and you slip on a banana peel and you scrape your knees. Your outer fortress has been breached and the pathogens are just flooding in like orcs through Helm's Deep. Banana factories are very dirty places. Now your body wants to contain the spread of pathogens, clean up the mess, and get healing as quickly as possible. So it cues up your inflammatory response. This is basically an internal fire alarm, only it uses chemicals instead of sirens to get the message across, and instead of smoke and fire, you sense redness, swelling, heat, and pain. For example, in the event of injury, specialized mast cells in your connective tissue send out histamine molecules, and histamine is great at calling in the cavalry. For one thing, it causes vasodilation, which creates redness and heat at the site of the injury. Now, those things might freak you out a little, but they're actually signs of healing. The increased temperature, for example, ratchets up the cell's metabolic rate so they can repair themselves faster. Meanwhile, histamines and other inflammatory chemicals also increase the permeability of blood vessels, causing nearby capillaries to release protein-rich fluids. This causes swelling, which again is actually a good thing because that leaked protein helps clot blood and form scabs, while the lymphatic system sucks up and filters that extra fluid, cleaning it up before putting it back into your bloodstream. And of course, like chum to sharks, an inflamed knee is also going to attract a bunch of local phagocytes, which find it easier to escape your now leaky capillaries, and lymphocytes that are also flowing freely, helping to destroy pathogens while also cleaning up dead cell wreckage. Don't forget, during all this, the neutrophils have been doing their best, but they were the first wave to arrive, so by this time, they're starting to die in heaps. They're triggered when the injured knee skin cells release chemicals that begin leukocytosis, the release of neutrophils from the bone marrow, where they're made, into the bloodstream. To attract the neutrophils to the damaged area, inflamed endothelial cells and the capillaries send out chemicals that act like homing devices. And when the neutrophils arrive, they cling to the capillary walls near the injury, flatten themselves out, and squeeze through the vessel walls to get to work. Your big monocytes eventually roll up to the battle and transform into hungry macrophages, replacing the first line of now dead neutrophils and basically just eating up any lingering enemies and then cleaning up the carnage. Now, all this works pretty well in most circumstances, but you may have noticed with a more major injury or an especially nasty virus or infection, sometimes your local troops get overrun. When white blood cells and macrophages run into more foreign invaders than they can handle, they let loose pyrogen chemicals that tap the hypothalamus and raise your body's thermostat, calling in a systemic fever to burn everything. The resulting temperature rise increases the metabolism of your cells so they can heal faster, and it also tells the liver and spleen to hold on to all of their iron and zinc so those things can't contribute to bacterial growth. But even then, sometimes you find yourself facing a more formidable foe. That's when you call in the specialists, your adaptive immune defenses. And to learn exactly how they save the day, you have to watch next time. But for now, you learn that your immune system's responses begin with physical barriers like skin and mucous membranes, and when they're not enough, there are your phagocytes, the neutrophils, and macrophages.
Right. So this is a great channel on YouTube that once you're doing biology and biochemistry, I find the graphics are really good and it helps you to visualize the concept. But it just reiterated what I said about the innate defenses of our system. So let's go to the next slide, which is the complement system. That is something that we, like basic immunology, you would usually just brush past it, like you would not make mention of it. But since I did it in my courses, I just wanted to show it. But the complement system is essentially some small soluble proteins. And what they do, they're activated for different pathways. So like yesterday, we were speaking about the monoclonal antibodies. In this case, if an antibody is bound to like a pathogen and it neutralizes it, what happens is like a one complement system, like I think the one complement protein, sorry, could recognize the FG part of the antibody and bind onto it, and then it would activate another one, and then another one, like a cascade, and then that would eventually lead to more of them becoming activated. So it's like almost like, I'm not sure if you guys have ever done blood clotting, but the way that there is a cascade in downstream cascade of activation, but that is essentially what happens in this case. But the cool part about it is that you form a MAC, which is like, it literally forms a pore inside of the the pathogen. So think of it as it forms a channel, like they come together and they form this channel within the membrane of the pathogen, and that channel allows for water to enter and essentially cause the pathogen to burst open. So because the pathogens are not like plant cells that have a cell wall, so yeah, they literally form a channel and that causes cell lysis essentially. Or they could also cause more macrophages to come onto the scene. But it's pretty crazy that these proteins could literally form a channel inside of the membrane of a pathogen, which leads to water entering and the cell to burst open. Yeah, so that's a complement system. Obviously, I give you a very um, brief explanation, but it's very much more complex than this. But yeah, I just wanted to share it with you. The other thing I wanted to share is a video of just showing how macrophages how they undergo phagocytosis. So let's just play this. It's a bit slow on my end. Let's see. Chance. Right, so this was just showing you just a mode of action of um, macrophages. So they have like almost finger-like protrusions outside of the cell and just go around. There's no specificity. They just go around and just engulf as many bacteria or any other pathogen that's in their path. And what they would do is after they destroy it on the inside, they have the capacity to display them like the antigen onto their surfaces, and what that would do is then lead to the activation of our adaptive immunity, where our B cells and our T cells would come into play, and then we could start producing antibodies against that bacteria. And that's just how our immune system would usually fight. So they work in tandem. So before I go on to the adaptive immunity, I just wondering if there are any questions. Okay, no questions. Pretty interesting, but I don't think it's difficult. It's just a lot. So yeah, we could go into the adaptive immunity. So the adaptive immune response is more specific and sophisticated defense mechanism, and it develops over time. It is activated upon exposure to specific pathogens and has memory, like I was showing you earlier. So it allows the immune system to respond more efficiently upon subsequent encounters with the same pathogen. So because it has a memory, you get like a quicker response because we've already encountered that pathogen before. And the key components of the adaptive immunity is the B cells, the T cells, and the memory cells that they produce. So this is a slide just showing comparing the activation of B cells and T cells. The adaptive immune response is essential for long-term immunity to infectious diseases and is the basis of vaccination. Vaccines expose the immune system to harmless versions of pathogens or like parts of pathogens, and that leads to the development of memory cells and immunity without causing disease. So we'll talk about vaccines later down in this presentation. But for now, let's just get into the activation of a B cell. So a virus affects a healthy cell. The cell is affected. And when the cell is affected, they usually, so all our cells, we have like an MHC complex that allows us to recognize self. So that's how our immune system is able to differentiate between what is foreign and what is like yourself, because you have that unique protein on the surface of your cells that says self. When viruses affect our cells, we usually either we lose that antigen, like that self antigen, or it's altered. So now your body recognizes that this is not right, something is wrong. And that is what our B cells would recognize. And when our B cells recognize that, it begins to release cytokines. So we start shooting out cytokines. These are like the interleukin 6, interleukin 1, several of them. I won't even go into it. But I have a slide showing you just the amount of cytokines that exist in our bodies today. But yeah, we release those cytokines and then our B cell becomes activated. And when it's activated, well, this slide doesn't show it, but it becomes activated with the assistance of a T cell called a T helper cell. And then that would also release more cytokines that would cause the B cell to become mature. But anyway, after the B cell is activated, you get memory cells, and then you get more mature B cells. And when the B cell become mature, it turns into a plasma cell. And I said this earlier, but the role of a plasma cell is just to produce antibodies. It just produces antibodies. Now, the thing is, it does not produce random antibodies. It produces antibodies that are specific to this virus. So the antibodies will look for this virus, surround them, and they would neutralize them by either binding onto them, or there are different modes of actions for antibodies. But after you form that antibody antigen complex with the pathogen, it makes it easier for the phagocyte to swoop in and basically behave like a garbage collector. So it just swoops in and just engulfs them, like just collects them and breaks them down. So that's essentially how B cells work. Obviously, like I said, it's way more complicated, but we want to go in so much depth. T cells, on the other hand, are so you could think of T cells as like the soldiers. Like if you look at war, they would be like the soldiers that actually go in and fight like by themselves. But in regards to B cells, they're like the archers. So they stay from a distance and they throw antibodies, which in this case would be like arrows at the pathogen. But T cells are more like hands-on with the pathogen. Like they literally interact with the cell themselves. So you could look here. When the virus infects the cell, the T cell recognizes the antigen that is on the cell, and then it starts releasing again cytokines to activate more T cells. These replicate, and these would cause the degradation of the infected cell. The way it does that is when it binds onto the cell, it releases um, some how would I term this? But it releases granzymes, and what they do is they make the cell more permeable, and then it also releases perforin. So granzymes makes the cell permeable, and then perforin will enter the cell, and it leads to gene expression, and basically induces apoptosis, which is natural cell death. So it would literally cause the cell to commit suicide. So after it binds to the cell, it would cause them to commit suicide, and then they die off. But like I said, with B cells, they release antibodies. You won't see a B cell binding onto an infected cell and causing it to just die by itself. It just produces antibodies, and obviously both of them would produce memory cells. So that's how we would achieve immunity. So if we have memory cells and this virus were to affect the cell again, and our memory cell recognizes the virus, immediately would start it would start differentiating into plasma cells and start loading out antibodies. And the thing with the secondary response is that we would produce way more antibodies than in the primary response. So that's why you find that if you encounter one, for example, if you got infected by one version of the flu, and later down the line, you would probably not be infected again with that same flu. Maybe you would get it contracted, but you won't. The symptoms that you would face would not last as long as when you initially got it. If you got the flu twice and the symptoms were as severe, then more than likely you were infected with a different strain. And like we spoke about yesterday, influenza is one of the viruses that could mutate very easily. So you find that you have various strains producing quickly. So now we're going to go into the phases of the adaptive immune response. So we'll start with the primary. So it begins with activation. So upon exposure to the pathogen, the antigen presenting cells, so that could be like your macrophages or your dendritic cells. They would show the pathogen's antigen on the surface and then present these antigen to like a helper T cell, like I was saying earlier, and that activates your B cells, in some cases your cytotoxic T cells. So in case you didn't realize, T cells can be branched into, well, yeah, I showed it on the slide earlier. But yeah, T cells branch into cytotoxic T cells and T helper cells. T helper cells interact with B cells to activate them. And cytotoxic T cells are those soldiers that literally go in and fight with the pathogen. So once a B cell is activated due to the stimulation from a T cell, it proliferates and differentiates into plasma cells. And then the plasma cells are those that produces the antibodies. The antibody production in a primary response, it takes some time for B cells to mature into T cells and produce sufficient antibodies. So that's why in a primary response, it's like it lasts longer than a secondary response. Because it takes some time for them to become mature and then produce enough antibodies to destroy the pathogen. And then you have immune memory. So that's when you produce the memory cells for both B cells and T cells. And these remember the pathogen, and these don't die off easily. So for example, I know in the Caribbean we do this, we take different vaccines at a young age. And that's why a lot of us have like a scar on our left hand. I can't remember which vaccine it is. I think is it for tuberculosis? I'm not sure, or is it, I can't remember which virus we got this shot for. But yeah, anyway, we got this vaccine, and this vaccine would have triggered our body to produce memory cells, and we would have achieved immunity for the rest of our lives just from taking a vaccine when we were babies. So that's to show you how long memory cells stay in the body. Obviously, all viruses or all pathogens are not the same. So in some cases, when you take a vaccine, you would have to take it more than once because they're able to replicate and mutate. And these things are so specific that there are different antigens. So that's why when we were in the pandemic, people were wondering, why do we have to take booster shots? Why do we have to take another vaccine? And we thought that it was a ploy by the government to put chips into you. But the reality is that a lot of us are ignorant on the fact of how our body works and how our immune system works. So I'm hoping that now participating in a program like this and doing your own reading, you're able to engage the community when they're spreading those propaganda, and you're able to clear the fog that's in their brains. But this is an example. I just wanted to show this slide of the different cytokines that are in our body during like an immune response. So you can see, I'm not even sure where to start, but we can start with the macrophage. Yeah, we can start with the macrophage here. So the bacteria affects us, and then the macrophage will start producing a whole set of cytokines. So TNF alpha, interleukin 14, interleukin 6, and then that would cause fever. It could then produce interleukin 8, TNF alpha, that would then cause the activation of neutrophils. So it just shows you the different the start points from just a macrophage in the initial infection, and then you lead to the activating of a T cell, and then T cell, you get B cells, and you get plasma cells. So it just shows how interconnected our immune system is, and the way they communicate is through these cytokines. So interleukin 3, interleukin 6, all these are known as cytokines. So this is how a typical immune response would occur in your body. Yeah, it's very complicated, but at the same time very interesting on how all these white blood cells interact. But now is the secondary phase, so that's when you get, so you've gotten the infection, it's already you no longer have symptoms, but in the next few months, you encounter that same infection again. What happens? So in this case, you already have memory B cells and memory T cells. And what they do is that they would quickly recognize the familiar pathogen antigens, and then that would lead to like a rapid and powerful immune response. So at this point, your memory cells would respond faster, more efficiently than when it was just a naive B cells when it wasn't activated, and they would quickly differentiate into plasma cells, and these plasma cells would produce like high levels of antibodies that are specific to that pathogen that affected us, and there would be a higher concentration of antibodies compared to the primary response. So if you ever did if you were to take a titer, like take a test to find out the amount of antibodies you had in a primary response versus a secondary response, you'd see that in the secondary there's way much more. So antibody production, the secondary response generates more significant and sustained increased antibody levels. So this results in a more effective elimination of the pathogen, preventing it from causing diseases. So I'm not sure if you guys have ever experienced this, but sometimes you get a sore throat and you're thinking, oh my God, like I'm going to get the flu again. But then the next day you're feeling fine. It could just be that, yeah, you encountered the flu, but your memory cells were so quick and efficient at clearing it up. And something that is very important with your immune system is your diet. So that is always good that you eat properly to have a very effective immune response. And also if you're not immunocompromised, that's also a factor. But the secondary response further strengthens the immune memory. Memory B cells and memory T cells are generated in even larger numbers, enhancing long-term immunity. So this is essentially stating that you would create more memory cells, but these would like stay for more and like be ready for another attack. So what this shows is that the more you encounter the same pathogen, the more your body gets used to it and the more your body can cope with it. And so eventually, if you do get an infection, you literally feel no symptoms. And this is a table just summarizing the primary versus the secondary response. So this is where you begin with a naive B cell. So this B cell is not yet activated, it's just waiting to become activated. When it recognizes the pathogen and it becomes activated through the help of a T cell, you then get plasma cells being produced, and these would produce antibodies. Now, I didn't go into the different types of antibodies. There are different types of antibodies, but these are Ig, IgG, and IgM. Those are usually the antibodies that are produced in the primary response. But yeah, these plasma cells don't live long, but you would also be producing memory cells. But when you get a second infection with the same antigen, the same pathogen, you get a more rapid. So if you look at the gradient of this graph, you realize that it's way less steeper than this one. This one is steeper. That's because you get a quick response and then you produce more plasma cells, and these would pump out antibodies. And the antibodies that's produced in the the secondary response is IgG and not IgM. So these are done in medical labs as well to see like if someone was ever tested for, if they've ever encountered a disease. Because like if you just did, for example, so if you just contracted a virus and your immune system is currently producing antibodies, if you do a blood test and we realize that you have IgM, then that means that this is the first time you encountered that virus. But if during your infection you're producing only IgG, then you have some form of immunity, some form of memory cells, because you're only producing IgG and no IgM. Yeah, but as you could tell, the amount of antibodies is way higher after the infection subsides. Because if you look at it here, this is a graph. So if you look at it here, you would see that it's way higher than in this case. Yes, you produced antibodies that killed off the virus or the pathogen, but the levels are still low compared to on the 30th day in a secondary response. And you could assume, you could infer that if this person was infected again with this same antigen, this same pathogen, that this would be steeper and the amount of antibodies would again be higher. So that's what I said. Like that's why I'm saying, like if you continuously get affected by the same pathogen, you can see how difficult it is for it to survive in our bodies. So now we're going to vaccination. A very interesting. Oh, before we go into vaccination, any questions before we get into that? I guess I don't see any questions. But yeah, going to vaccinations, and that was a very interesting concept, especially during the pandemic. I remember at the time I was in my first year at university, and I was getting so many questions from people, and I felt like I needed to answer. Like there was so much controversy going on the internet, people spreading stories about how vaccines work and why you should not consume vaccines and how vaccines would change your genome. And there are so many speculations around vaccinations that it was really crazy, but it was also a great opportunity to just read more on how vaccinations worked. And well, people heard the fact that we were using mRNA vaccines, and that meant that it would go into our genome. And yeah, but I'm sure that you would have heard the talks on social media and different forms of media. But in case you didn't know, I'm hoping that I could clear any confusion today. But vaccinations, also known as immunizations, are medical interventions that stimulate the immune system to build immunity against specific diseases. They play a crucial role in preventing the spread of infectious diseases and safeguarding public health. So how do vaccinations work? So we introduce antigens. So the vaccine could be a weakened or an inactivated pathogen, or it could even be pieces of a pathogen. So just the antigen, and that triggers an immune response. Now the immune response would be our immune system recognizing these antigens as foreign invaders, and they would produce antibodies to fight them. And like we just went through the whole process, when our B cells are stimulated and they differentiate into plasma cells, we also form memory cells. So after we create those antibodies, we'll also be forming memory cells, and these memory cells will remain in our body, and that's how we establish immunity. So that's how a vaccine works. So we introduce a very weak or parts of an antigen, or in the case of Moderna, what they did was they introduced the mRNA of a virus, and that is what triggered an immune response and helped us to build up um, memory cells and immunity. So there are different types of vaccines. You could have live attenuated vaccines, you could have inactivated vaccines, subunit, recombinant, or conjugate vaccines, or you could have viral vector vaccines. These are all the different types of them, and vaccines started from, I believe, the 18th century. I believe was the first vaccine that we created. I might be, I think either the 18th or I think it's the 18th century. Okay. So vaccine safety and misinformation. One thing that people don't know is that there is extensive testing being done in vaccinations. We don't just release vaccines into the public. There are several stages of clinical trials that have to go. So vaccines undergo vigorous testing and clinical trials to ensure their safety and efficacy before approval. And we don't, well, scientists don't start um, testing like a vaccine in a clinical trial on humans right away. First, it goes on to like rodents, for example, different animal models, and then when we feel like the efficacy and the results are high enough, then we could move on to human clinical trials, and they probably go through years of human clinical trials because before we could say, okay, cool, it could be used on the public, it could be released to the public. And we were something you had to explain to a lot of people during the pandemic was the point was, how comes the virus came out in 2019 and by 2021 we already had a vaccine. But the thing is that coronavirus was not a new virus. There are several members of the coronavirus family. There's MERS, that's also part of the family, and these viruses had vaccines already. So it was really now that were vaccines that were already approved. So it was really just tweaking those vaccines to suit the coronavirus that was affecting us to this date, and that's how we were able to release a vaccine in short such time. But a lot of people would not believe that, think it's a ploy against humanity or big pharmaceutical companies trying to get more money. So there's a lot of controversy around it. But another thing about vaccines is addressing concerns. So misinformation about vaccines can lead to vaccine hesitancy. Health organizations actively address concerns with evidence-based information, and that's a very key one because a lot of information is being spread, like I've been talking about recently. Various side effects. While vaccines have side effects, the benefits of a vaccine outweigh the risk. Sometimes you would get obviously a lot of things with our system, and genetically different people react to treatment differently just because of the way our genome is structured. Everyone is unique, so you find that different people would encounter various side effects. But again, during the clinical trials, we would go through those side effects and make sure that they're not severe. Yeah. So now I wanted to touch on some autoimmune diseases since we're doing immunity. And if you're part of my class yesterday, you would know that I tend to bring in as diseases or disorders up as often as I can. Because understanding a disease means that we could find a way to get a cure. So if we know what pathways are involved in a disease or what our genetic makeup is linked to it, then we could find out like how we could make changes to, basically cure the disease. But today we'll do autoimmune diseases. These are a group of disorders where the immune system designed to protect the body from foreign invaders mistakenly attacks healthy cells and tissue. This can lead to a range of debilitating system, sorry about that, and chronic conditions. So mechanisms of autoimmunity, so the breakdown of tolerance. So normally the immune system recognizes the body's own cells as self and avoids attacking them. In an autoimmune disease, this self-recognition breaks down. So earlier I was speaking about the fact that our immune system is able to tell foreign to self because of a self antigen that we have on our cells. So all your cells in your body right now have that antigen. And that tells our immune system that this is part of you and it should not attack it. But if by chance, for some reason you were to lose that that antigen, that self antigen, then right away your body does not, your immune system does not have a thing like saying like, okay, I've seen you before, I remember you. No, from the time your body, your cell does not have that self antigen, you would look like you could seem like a virus or some other pathogen, and it would begin destroying it. There's also the case of where you produce auto antibodies. So immune cells produce auto antibodies that target healthy cells, leading to inflammation and damage. So when your B cells are being formed, we didn't go into it in this lecture because it's a bit more complicated. But when you're forming B cells in like the bone marrow, there are some cases where you could form a B cell that recognizes the antigen of your body. So that's what we call an auto antibody. So that B cell recognizes your self antigen. And in that case, it would produce antibodies against that antigen, meaning that it would affect you. So it will attack you. And that is seen in diabetes type 1, where your B cells will start producing antibodies against your cells in your pancreas. And then, so if you're breaking down cells in your pancreas, you're unable to produce insulin, and then you would develop diabetes. So that's why well, right now, a lot of research is being done into linking type 1 diabetes to your genetics, because we're trying to understand what codes for those B cells to start producing antibodies against the pancreas, cells of the pancreas. And yeah, so right now I think they've discovered about 3 or 4 genes that are linked to type 1 diabetes, but it's not concrete as yet. So a lot of research is still being done into identifying the specific genes that lead to the development of type 1 diabetes. But it's something you could always read upon. It's very interesting, like I said. So examples of autoimmune diseases, like I said, type 1 diabetes, the immune system destroys insulin producing cells in the pancreas, and that leads to high blood sugar levels. It's also seen in rheumatoid arthritis, which is a chronic condition causing joint inflammation, pain, and swelling due to immune system attacking the joint tissue. And multiple sclerosis, where the immune system attacks the protective covering of the nerve fibers, disrupting communication between the brain and the body. So these are some disorders that are seen in, especially in the Caribbean region. Um, so yeah, a lot of research is being done into that. And triggers and risk factors. So genetic predisposition. Certain genes increase the susceptibility to autoimmune diseases. So like I was saying, right now a lot of research is being done into understanding type 1 diabetes. Environmental factors. So infections, toxins, and other environmental triggers can activate autoimmunity in genetically susceptible individuals. Humoral influence, where hormonal changes, particularly in women, are linked to the onset or exacerbation of some autoimmune diseases. So you could tell from environmental factors and genetic predisposition that your genetic makeup does play a role in the development of autoimmune diseases. So that's why I believe that genetics is really the future. If we could understand how these things work and the link between them, then we could literally work towards curing a lot of diseases or forming remedies that could basically seize um, the how they affect us. But that concludes my presentation on immunology. I tried to squeeze as much as I could for this presentation. Um, For those of you who weren't here yesterday, we had a game called Kahoot. Just to summarize what we just learned today. So as I set that up, I'm just opening the floor to any questions. Okay, so in the event we have no questions, I'm going to share the game. It's called Kahoot. If you've never played it, the way it works is you would go onto your phone or your laptop. I think your phone is better. And you would go to this website. So let me just turn off the volume. But yeah, you would go to this website, kahoot.it, and you would put in this code, or you could scan this QR code. It would bring you directly to the website. So I will put the URL in the chat. I could also, yeah. So what you do is you go to this website, enter a name. I see that we have 2 individuals already. How many people? Okay, so missing 2 more. So you can either scan this code from the screen or go to this website. So the way the game works is it's multiple choice. So you're competing against each other. You get points for the correct answer, and you also get points for how fast you answer the question. So on your screen, you would only see the shapes and the color. So you'd have to look at my screen to see the question and the options. But on your screen, you just click on the answers. So I give the others 2 minutes to join, and we will start off after. Okay, so I guess it's only Crystal and Giovanni, I believe. Let me see. Yeah, Giovanni. So Giovanni won yesterday's code. So let's see if we could reclaim, if we could continue it, or will Crystal take the crown? So we're going to start. So get ready, guys. So the first question, what is the primary role of the immune system? Digestion of food, transportation of oxygen, production of hormones, defense against pathogens. So on your screen, you would, yeah, so both of you got it correct. That is correct. So right now, Crystal is in the lead. Next one, which cells are responsible for producing antibodies? Is it B cells, T cells, natural killer cells, or red blood cells? Yeah, that is correct. It's your B cell, particularly your plasma cell. Crystal is still in the lead, but the gap is really close. Next one, what is the main function of antibodies? Do they destroy healthy cells, stimulate inflammation, enhance oxygen transport, recognize and neutralize pathogens? 3. Yeah, so both of you did get it correct. They recognize and they bind onto the antigens of the different pathogens, so neutralizing them, making them useless. So Giovanni has passed Crystal, I guess, because he answered the question faster. So you have to keep that in mind. Next question, which immune cell type is responsible for remembering previous pathogen encounters? So T cells, macrophages, memory cells, B cells. Yeah, that is correct, your memory cell. So So Giovanni is still at the front. What is the purpose of vaccines? Four, three, two, one. So the one person answered it, but it's to prime the immune system against specific pathogens. So Crystal got that correct. Giovanni, I guess you didn't answer the question. So now you have to catch up. There is at least a 600 point gap. What is the role of macrophages in the immune system? What is the role of macrophages in the immune system? Yeah, that is correct. Engulfing and digesting pathogens. I showed you all the video of just how they work. So Crystal is still in the lead. 3 questions left. Which immune cell is primarily responsible for attacking virally infected cells? Now, I don't think I said that specifically, but I'd like to see who gets this one. Yeah, that is correct. T cells. Next question. What is an autoimmune disease? What is an autoimmune disease? Okay, so someone said virally induced disease. That is not correct. It's when the immune system attacks itself. So like we just spoke about, when your body could produce antibodies against itself and destroy um, our own cells when it does not recognize self. So example, diabetes type 1. So with that, Giovanni has passed Crystal. A few more questions left. 2 more questions. The secondary response is quicker than the primary response. That's a true or false. Yeah, that is true. And I show that with the graph. And last question, true or false, the skin is part of the innate immunity. A very simple question. Don't expect you all to get this wrong. So is the skin part of the innate immunity? Yeah, that is correct. So it's time for the podium. Well, we have no third place, is either second or first. So in second place, we have Crystal. And for back-to-back sessions, we have Giovanni winning our Kahoot. The gap was really close though. So, but congratulations, Giovanni, on winning this. Yeah, but this is it was a very nice way to summarize all the content that I did. Right now, we will be taking a 30-minute break. So we come back 5 past, well, for me, it's 5 past 1. I think for you, it would be 5 past 12. And then we begin our last session of the day, which is biotechnology in medicine. So see you guys in 30 minutes.